Imagine taking a patient's own immune cells, outfitting them with a specialized biological radar, and sending them back into the body to hunt down cancer. These are chimeric antigen receptor, or CAR, T cells. In liquid cancers like leukemia, this approach has been highly successful. These engineered cells can track down the target and clear out the disease entirely. But when doctors deploy these exact same hunter cells against solid masses, like kidney or pancreatic tumors, they fail. The T cell's weaponry remains fully functional, but the solid tumor's biological conditions create a sensory gap that renders the standard hunting strategy ineffective. Unlike liquid cancers, a solid tumor isn't a uniform mass of identical cells. It's a chaotic patchwork. The cells display varying amounts of specific surface proteins, like one called CD70, that our engineered T cells are programmed to track. This diagram illustrates a typical solid tumor. The dark red cells are loud. They are fully coated in the CD70 target. But right next to them are these light pink cells that appear to be completely empty. During a standard CAR T cell attack, the engineered cells follow the left pathway, easily destroying the loud, dark red targets. But the seemingly empty pink cells survive and multiply rapidly, driving aggressive cancer recurrence, known as antigen escape. By wiping out only the most obvious targets, standard CAR T cells act as an accidental evolutionary filter. They clear out the weak, leaving behind a stealthy, invisible enemy. Researchers started to wonder, what if those escaping light pink cells aren't actually empty? What if they still have the CD70 target, but they're just hiding it? Cells have a biological trick called epigenetic silencing. When a cell doesn't want to use a specific piece of its DNA, it tightly coils that chain up, physically restricting access to it. At the bottom right, tumor cells use EZH2 to lock down the CD70 gene, creating a CD70 ultra-low state. The red X shows why standard therapy fails. Normal CAR T cells need a loud signal to attack, remaining completely blind to this microscopic whisper. The solid tumors didn't lose their target. They just lowered their profile right below the mechanical radar of our living drugs. If the tumor turns down the volume, the solution isn't to look for a different target. The solution is to build an antenna that can hear a whisper. This is the HLA-independent T cell receptor, or HIT. Looking at the structures on the right, it is a newly designed cellular antenna, built specifically to catch what standard CARs miss. This chart shows the difference in activation sensitivity. The standard CAR activation line sits high up, letting low signals slip by. But the HIT receptors line is drastically lower, hyper-calibrated to trigger an attack even when detecting ultra-low, previously invisible traces of CD70. Instead of searching for new proteins to target, the researchers lowered the sensory threshold of the cells themselves. To test this, scientists set up a rematch. They deployed the new hypersensitive HIT cells against the exact same patchwork solid tumors that had easily defeated the standard therapy. Following the pathway on the right, the HIT cells detect both the loud and the whispering ultra-low cells. Because these ultra-low cells are hunted down alongside the rest, the escape route is cut off, resulting in total tumor eradication. You can destroy a solid tumor if you close the evolutionary loopholes the cancer relies on to survive. But making a drug this sensitive introduces a serious medical risk. If these new T cells can attack cells with barely any CD70, won't they accidentally attack healthy organs? Mapping healthy adult tissues reveals a critical safety margin. In normal cells, the CD70 gene is physically locked by epigenetic markers, keeping expression at a true zero. This chart compares the three states. Healthy tissue sits at absolute zero, remaining safe from attack. The HIT cells require at least an ultra-low whisper to activate, bypassing healthy cells while eradicating the cancer. These findings indicate that what was once considered untargetable was actually a failure of detection. To overcome resistance in solid tumors, we may not need to discover new targets. We need receptors tuned to a much lower threshold.